Thank you so much. I also been told that I have 20 minutes instead of 30, so I will try to go very fast. But first, I would like to thank you for this opportunity of being here. It's a great honor for me to be here in Pakistan to present this topic. So thankfully, in this brilliant session, there has already been quite some talk about Fowl Adenovirus, so I don't have to spend too much time in the introduction. You already know that it's a non-enveloped DNA viruses, and there are 3 diseases associated with these viruses that are affecting the poultry industry: Inclusion Body Hepatitis, or IBH, Hepatitis-Hydropericardium Syndrome, or HHS, and Adenoviral Gizzard Erosion, or AGE. This is a phylogenetic tree based on the partial exon gene, and these are the 5 species of Fowl Adenovirus. Now, the taxonomy has actually very recently been updated, so they are not anymore called species A, B, C, D, E. They have Latin names now, but because most of the literature and also most of the people still refer to the Fowl Adenovirus with the lettering, I will still doing so in this presentation. So the adenoviral gizzard erosion is associated with serotype 1 that belongs to species A. Hepatitis-Hydropericardium Syndrome that has been told that we actually first described here in Pakistan in 1987. It's associated with serotype 4 belonging to species C. And then there is Inclusion Body Hepatitis that has a more diverse etiology. In fact, this is associated with serotype 2 and 11 from species D and 8A and 8B from species E. Here you can see the genome of the viruses. In the central part, there are the genes coding for the major structural proteins. Well, the terminal region are more genus-specific, but I would like to draw your attention to the fiber gene because it's important for our talk to know that species A and C have 2 different genes codified from 2 different fibers of different length that you see associated here for each apex in the penton base, whereas the other species only have 1 gene codified for 2 identical fibers. You already know about the disease. I don't have to go into too much detail here. These are Hepatitis-Hydropericardium Syndrome and IBH are quite similar in pathogenesis and immune response. Their main host is the young broilers. And as the name says, for HHS, major finding is hydropericardium hepatitis. For IBH, there are these very specific inclusion bodies that you can see in the nuclei of the cells of target organs, especially in the liver. So this is a map in the world, and this is how the diseases IBH and HHS are distributed. So in the pink areas, you see where IBH has been reported, where the stripes is where HHS. And while you can see IBH has been reported for basically all the continents, there are still some areas that has not reported HHS to this day, which include Europe and most of North America. But HHS is a huge issue in many areas, especially in Asia. Here in Pakistan is a big issue in China as well. So what is the vaccine situation regarding what is commercially available? There are licensed vaccine FAdV 4 killed in Asia. Actually, during this conference, I found out there are actually killed vaccines against serotype 8 and 11, for example. And there are licensed vaccine in Oceania, and there are live vaccine against IBH. But some places, including Europe and North America, relies on autogenous vaccine or homologous vaccines. In the field, it's not uncommon to find mixed infections and shift to outbreaks from one serotype to the other after introduction of vaccination have also been described. In this, you can see the case reports in scientific publication per year, starting in 1963 when IBH was first described, up to these days. And as you can see, this is not an issue that is going away. This is an emerging issue. I like this picture very much. I took this from a review from South Korea, from Professor Mo, because here you can see the shift in the major serotype causing outbreaks from 2007 and how the situation changed a lot through the years until 2021. This is a demonstration of the mosaic of how plastic the situation is in the field. And this tells us that if we want to design an effective immune strategy to tackle this disease, we have to deal with the problem of a broad spectrum protection. The ideal vaccine needs to be as broad protective as possible. Obviously, people are aware of that. There is a lot of research going on, more than 70 publications, and they're especially concentrated in the last few years. This is why this is also an honor to be here in Pakistan talking about it. As Professor Naeem already said, this is the first country that actually reported vaccines against Fowl Adenovirus. So really, without you, this talk won't be possible. So subunit vaccines have already been described. In the case of Fowl Adenovirus, the formulation are based on recombinant capsid proteins. And this concept has been borrowed from studies that are unknown for the virus, such as ADS-V virus and HEV. But these adenoviruses only have one fiber, as opposed to the situation of Fowl adenovirus, as we have seen. So the antigenic composition is a little bit more complex in this case. This is a list of what has been described in the literature. We will go through it, don't worry. What I want to stress is that this efficacy has been demonstrated against HHS and IVH. So we are not going to talk about adenoviral gizzard erosion. And the reason is that in this case, there is no role of systemic protection. And we know that because chicks that have maternal antibody against FAdV1 are not protected against the disease. So the reason is because this disease affects mostly the gastroenteric tract, and we need a local immunity. How to reach local immunity is to give oral vaccination. So in this case, only live vaccines has been reported at the moment through the administration of apathogenic or attenuated strains. So let's dive into what a classic in vivo protection study looks like. So you need to express your protein of interest, and then you vaccinate the chicks with the formulation. So we're talking about the subunit here. So an injection is necessary. Usually it's intramuscular or subcutaneous. And then you divide the chicks into groups. We always opt to have them in isolators, so they don't, the virus cannot obviously exit the group. You always need a vaccine plus challenge group, so chicks that have been vaccinated and then experimentally infected, challenge control, only infection, and a negative control. No vaccine, no infection. These are the very basic groups that you always need. During the study, you monitor the human response by taking frequent blood samples, and then you can perform ELISA and virus neutralization tests. At some point, the chicks are challenged with the virulent Fowl adenovirus, and then in different days, there is a sequential euthanasia of parts of the chicks in the groups, and then you perform your necropsy, you take all the samples you need. Sorry. In this case, you also have the chance to collect blood and organs to monitor the cellular response through flow cytometry. Not many people do this, but now it's getting more and more interest is being developed in this area, and I'm a huge advocate to always check the cellular response as well if you have the chance. So let's start with Hydropericardium syndrome here, and my plan was to start with the pentomase, but Professor Raham already actually talked extensively about this, so I will just briefly touch upon it. So this is the first subunit vaccine that has been described by SAHA in 2012, and it was quite effective. They saw development of ELISA antibodies and 90% survival. However, as we have already seen, subsequent studies reported that with the full phantom base, there was partial to 50% to no protection at all in some studies, despite the presence of the ELISA antibodies. And also, it has been demonstrated that there can be less efficacy compared to other FAdV4 subunits. Now, we have seen that with the truncated insertion domain, you can achieve up to 100% success. So this is a very specific case concerning the pentom base. So if you're interested here, I will refer to Professor Raham for this. And I will proceed with the exon. There are some studies also using the exon, the FAdV4 exon against HHS, although they reveal partial to no protection and also less efficacy compared to other subunits. Also, there is one study that used it and showed that there was no neutralizing antibodies production despite the presence of antibodies on the ELISA. So there is also one study that checked a minor structural protein that is the 100K. They found no protection there despite the presence of ELISA antibodies. So nobody else, again, went in that direction. So let's talk about the fibers. As you remember, there are two fibers, fiber 1 and fiber 2 for FAdV4. And for fiber 1, there are several studies that focus on this. And the protection has been reported only in the study that used a prime booster vaccination scheme. So they had a prime vaccination and then a few weeks later on, we have a booster vaccine. But in one study that had only one application, there was no protection with fiber 1. Also, there are conflicting reports about the presence of the neutralizing antibody. In one case, there were no neutralizing antibody. One other study actually showed that there were. You can see here, for example, in this case, before challenge here, no neutralizing antibodies. This is another paper, and instead they have a pretty good development. And probably the reason here is because you saw before what is the setting of the in vivo protection study, but there are always differences in the quantity of the protein that is used, the age of the chicks that are vaccinated, etc. And apparently for fiber 1, this is very important when it comes to the development of neutralizing antibodies. So fiber 2, spoiler alert, fiber 2 is the actual rock star of the subunits against FAdV4. In fact, this is a very nice study that was from my group, but before I joined, from the group of Professor Hess, and they use different FAdV4 subunit, fiber 1, fiber 2, the exon, and this is the challenge control, and these graphs indicates the mortality after infection. And you see that it was pretty high challenge control, similar in the exon, pretty high in fiber 1 as well, but very low in fiber 2. Actually, I will tell you, this was only one bird that died, and it was the only bird in the group that did not develop antibodies in this case. It is interesting, very interesting to me to know that here we have protection, but we did not have the development of neutralizing antibodies, although we do see antibodies on the ELISA. Later on, many studies have focused on FAdV2. It's consistently good, and somebody even checked Chen in 2018, the cellular immunity, and they saw a systemic increase of the CD4 T cells after vaccination. So what happens with IBH? So this is actually when I started my PhD, they told me you have to find a broad protective vaccination strategy against IBH. I said, great, sure, okay. Let's start with the pentom base, because the pentom base is quite conserved. You remember, in IBH, there is a more diverse etiology. The pentom base within species E, it's quite conserved compared to other subunits like the fiber. So we tested 2 pentom-based proteins from FAdVE, one belonging to serotype 7, one belonging to serotype 8B, and we challenged with a pathogenic 8B. And in both cases, no protection. No protection in the second case, we even waited because there was low immunogenicity on the ELISA. We applied a booster vaccination to ensure that there was a development of antibodies, but still, after challenge protection was not there. So my PhD did not go off a great start, although we do get some pretty good information. Maybe at the time, if I knew Professor Rehman, I should have talked to you before, but I was pretty depressed. And so we move on testing the fibers. Now, at the time, there was already one study that used the fiber from 8B, and they even tested the vertical protection, meaning they vaccinated the breeders with fiber 8B, and then they challenged the protein with a pathogenic homologous strain. Yeah? And in this case, the chicks were protected by the transfer of the maternal antibodies. They even had a look in the systemic cellular immunity of the breeders. After booster, they saw that there was an increase of the ratio between CD4 and CD8 cells. So what we did at the time, we wanted to tackle the broad protection, right? So we took fiber from 8A, we vaccinated the chickens, and then we challenged them with either virulent strain 8A and 8B. And what we saw is that the chicks were only protected against the homologous serotype. You can see here why, because there was a serotype-specific neutralizing antibody development. This is the challenge strain 8A, so they did have neutralizing antibodies before challenge, but not in the case of 8B. So they were not protected. So the information I got at the time from this was protected from IBH depends on serotype-specific neutralizing antibodies. In that case, we also did a pretty extensive investigation of the systemic cellular response after vaccination with fiber 8A. And what we saw is that the vaccine prevented a systemic drop of the B cells and also the monocyte macrophages that was caused by the challenge. So the solid line is the challenge, and you can see that was significantly low compared to the basal level, which is this one, whereas the vaccinated groups do not undergo such a pathologic drop. Instead, in the vaccinated, there was an increase of systemic CD4 T cells and also a modulation of pathological changes of the CD8 alpha T cells. So I said during my PhD, started with the penton-based, did not work. Started with single fiber, worked only homologously. So what to do now? So to tackle the problem of the broad protection, we decided to design a recombinant chimeric fiber protein that would merge epitopes from 8A and 8B fibers. So here you see the concept. So we have a full-length fiber. And in the terminal domain, which is this one we call the knob, we introduced a specificity switch. So from one side of the switch, we have identity of fiber 8B, and on the other side, we have identity of fiber 8A. This is how it looks like a little bit more in details. This is a 3D scheme of the knob, and this is a 2D scheme. Here you see the switch. So from one side, you have epitopes 8B, and then it switches and becomes 8A. That's how, more or less, how it looks like in a scheme. So we took this chimera, we vaccinated our chicks, and this time, so we challenged them with 8B, with 8A, and this time it did work. So we had cross-protection, we had cross-protective antibody, we saw that on the ELISA, and also neutralizing antibody against both 8A and 8B. At this point, we were like, okay, I can get my PhD, thankfully. So we decided to extend the concept of the chimeric fiber to see if we could include protection against HHS. So what we did before was merging epitopes from different serotypes but belonging to same species of Fowl Adenovirus E. What we wanted to do now is to merge serotypes from different species. And we took serotype 4 and 11, so species C and D, and we did the chimeric fiber for 11. And just because we saw before and we know by literature that fiber 2 for FAdV4 is the most successful one, that's what we selected for our chimera. So we took these, injected in the chicks, challenge with 4, challenge with 11, and once again, we were successful. But despite the fact that we did have ELISA antibodies, we did not have neutralizing antibodies. So this is a similar situation of what we have with the protection used by fiber 2 for FAdV4. You see here, no antibody against FAdV4. I unfortunately did not put the figure, but we did have very little antibody against 11 only in a couple of birds. But protection was there. So in this case, because we kind of expected something like this, we really looked into the cellular immune response. We published a paper with extensive results. Unfortunately, we do not have too much time here, so I will go directly into the highlights. So these are the B cells, the systemic, so in the blood and in the liver as well. And this dotted red, if you can see, is the vaccine group. The solid red is the challenge control. And you see, for example, in the blood, so systemic response 3 and 5 days post-challenge, there is a significant increase of the B cells in the vaccinated ones, but not in the challenge control. And in the liver, which is the major target organs as well, the vaccinated ones have a faster increase compared to the challenge control. So we see a certain priming effect of the vaccine. These are monocyte macrophages in liver and spleen. At the very early point of infection, 3 days post-challenge, we see that both the vaccine plus challenge and the challenge control, they significantly rise compared to basal level. But in the liver, immediately after, only the challenge control remains pathologically high, whereas the vaccine goes back to basal level. In the spleen, actually, both groups, again, they have a significant drop compared to basal levels, but by the end of the experiment, only the vaccinated ones went back to basal levels. So CD8, so cytotoxic T cells, we have a proliferation in the thymus in the very early stage of infection, and then significant increase in both vaccinated plus challenge and challenge control in the liver, so target organ, but once again, at the end of the experiment, only challenge control was pathologically high. And this is, in my opinion, the most interesting result in this regard, because in this experiment, we also had one group that was only vaccinated, so no challenge, and this is this fade blue that you see here. And we saw that in the spleen, which is also a target organs, there was a prolonged increase of the cytotoxic T cell. So without the infection, the actual infection of the virus, just with a subunit, up to almost 5 to 6 weeks after vaccination. Now, the one one developed from our group is not the only chimeric subunits we have seen also previously. There's a publication from 2021 of truncated fiber 2 of FAdV4 encoding exon epitope that was also successful, mainly depending on the dose. So with high dose, they were 100% survival. With lower doses, a little bit of less. And also they saw development of antibodies on the ELISA. And then more recently in 2023, there is also a group that took all the major subunits, so fiber 1, fiber 2, penton-based exon, and they did chimera, and they put them in different combinations, and they had different degrees of efficacy, you can see here. And the ones that produced the higher degree of neutralizing antibodies also had the highest survival, actually full protection, 100% survival. So in this case, there is a dependence of the development of neutralizing antibodies as well. The NOB alone in some cases has also been protective, and in 2024, Liu et al. Demonstrated that a fusion NOB of FAdV4 between fiber 1 and fiber 2 also granted full protection and also had development of neutralizing antibodies. More recently, there has been much more focus on recombinant vaccines, and these are recombinant FAdV4 vaccines incorporating subunits from different FAdV species. So this is also an attempt to tackle protection against HHS and IBH at the same time. It's an FAdV4 that also expresses fiber from serotype causing IBH. They did it with 8A, fiber 8B, and even fiber 8B plus 11, and they saw induction of serotype-specific neutralizing antibody, and therefore the species that were covered by these neutralizing antibodies were protected. For example, in this case, when they did not include the fiber FAdV8, there were not neutralizing antibody 8A, sorry, not 8. There were not neutralizing antibody against 8A and no protection. Lastly, there are also Fowl Adenovirus recombinant capsid proteins that are delivered through vectors. We had Newcastle expressing the fiber FAdV4, a fiber 2, that is delivered intramuscularly. This is a study from 2020, also granted full protection against both challenges, so NDV and FAdV4. Also, there are tested non-viral vectors. We have Lactococcus lactis, Enterococcus faecalis, and also Saccharomyces cerevisiae expressing fiber 2, or in the case of the bacteria, also exon of FAdV4. These are very interesting because they can be given orally. So oral administration, especially in the poultry management, is very important if it can be achieved. If you compare the studies, you see that some vectors perform better than others. For example, Saccharomyces cerevisiae and Enterococcus faecalis were better than Lactococcus lactis. And as we saw before, fiber 2 performed better than the exon. There was development of the antibodies on the ELISA, and one study even looked at the neutralizing antibodies. They were observed on Enterococcus faecalis expressing the fiber 2. This already leads me to my summary and conclusion. So Fowl adenovirus-related diseases have increasingly important economic impact on the poultry industry. Subunit vaccine based on recombinant capsid proteins are efficacious against HHS and IBH, and especially fiber-based formulations. And these recombinant chimeric fiber constructs that are retaining epitopes from different Fowl adenovirus serotype species can elicit cross-protective antibodies. We saw in many cases protection from HHS is not necessarily linked to the development of neutralizing antibodies. And recombinant fiber vaccines have also been shown to elicit significant systemic and local cellular response, although few studies so far have focused on it. Recently, formulation containing recombinant FAdV4 edited to express fibers from different species and serotypes have been described, and vector vaccines with via viral bacterial or yeast backbones expressing capsid proteins are also been studied. Further investigation of the immune mechanisms behind this protection are needed, especially when we don't have neutralizing antibodies. With this, I would like to thank you all once again for your attention.